Thank you very much, Stephen.
Thanks, Richard.
Is my slide visible?
Yes, it is. Thank you very much.
Excellent. So greetings to everyone. Thank you for joining today's webinar. I'll be sharing with you a summary of the skin-NTD situation in Ghana and where I think we are with diagnosis from where I see it from. So as you know, we are part of the big ecosystem, and the arrow on the map, the blue arrow, shows the location of Ghana. Ghana is a West African country bordered by 3 other countries. To the north is Burkina Faso, to the right Togo, and to the left is Cote d'Ivoire, and we have the Gulf of Guinea down there. So Ghana is pretty much what we see with the arrow blue pointed to. So I'll first begin by us reflecting on what getting a skin-NTD diagnosis would entail generally. So I'll look at it first from the process involved with initial assessment of the patient when they come to the facility. So first, the health worker will need to know how to examine the skin. They have to know the common skin diagnosis to be able to know what to suspect when a patient walks into the facility. If that is not there, you have an initial problem with achieving diagnosis for the conditions in the first place. Secondly, you have to look at whether or not you have a point of care test available, or you're going to have what we call a send-away sample, where the sample will be tested in the lab. In the case of the point of care test, the health care worker will have to know how to interpret the test. They have to know how to act on the result, and we have to also ensure that treatment is available for that test. Otherwise, there's no very much value of doing the test and where there's no care. So that is an issue to consider. And where there is a send-away test, where we have to get the sample to a lab outside the facility of the testing, then there are issues of communication and the fact that the patient has to return to receive the result and then possibly start treatment. So all of these are considerations to look at. And in many parts of the world, in Ghana, many of the communities, transportation and the road networks can be a challenge, as you can see on the picture in the screen. So the conditions in the skin-NTD spectrum are many. In fact, we have about 11 currently, as shown on the screen, ranging from Buruli ulcer all the way to the latest identified and added, that's NOMA. And of these 11, Ghana has 7. But before I go to the ones Ghana has, I just want to mention that we are also working in line with the rest of the world to meet the various roadmap targets set by WHO. And as you can see, several of the skin-NTDs are having their target as control, but there's one for eradication, yours, and Ghana has yours. And so we also hopeful that we can eradicate it. 3 of them are up for elimination at the various country level, leprosy, onchocerciasis, and lymphatic filariasis. So to Ghana specific, and I just presented these first 2 slides to just say that we acknowledge very much that Ghana is part of the global ecosystem, and interventions for diagnosis that are being discussed are first from the global perspective and looking at what is happening in the Ghanaian context. So in Ghana, of the 11, we have 7: Buruli ulcer, cutaneous leishmaniasis, leprosy, lymphatic filariasis, EOS, scabies, and other ectoparasites, as well as onchocerciasis. All of these are endemic in Ghana, and the National NTD Control Program is working hard at ensuring that these are all addressed under the umbrella of the control program and in collaboration with research institutions, including the Noguchi Memorial Institute for Medical Research. So this slide is a bit busy, but it just lists the various skin-NTDs with a focus on the ones in Ghana, the current sampling approach, and the recommended diagnostic tests. So for all the tests, the recommended diagnostic tests here are as recommended by the WHO. Ghana currently, I would say, has all of them in place, except a few. And so I'll just mention the ones that we are not actively doing as a country. So when it comes to scabies, the main diagnosis currently is clinical diagnosis. We are not currently doing dermoscopy or video microscopy, which are possible options. A lot of the diagnosis really is based on the clinical diagnosis. When it comes to Buruli ulcer, the mainstay is the PCR testing to confirm the bacteria using either swab samples from ulcers or fine needle aspirates when it's not ulcerated. For cutaneous leishmaniasis, although the microscopy and PCR can be used on skin samples, particularly or especially biopsy, Ghana currently is conducting PCR mainly at the research level. Routinely, we are not yet doing this test at the point of care. There are discussions to move to the use of microscopy at the point of care whilst we continue to do PCR at the research level. But that is not currently happening yet, and so we are working towards that. For leprosy, the slit skin smear or skin biopsy is what is recommended. Microscopy is what is being done largely. There are efforts to look at PCR or histology, but I would say that there are specialized facilities that are doing the slit skin smear in Ghana, and they are identified, but it's not across the country. So they identified facilities for that. For lymphatic filariasis, the main diagnostic test now is a microscopic examination of blood smear for the microfilaria. There are some rapid tests, diagnostic tests, but these are usually more for research, okay? And then for onchocerciasis, the mainstay is also microscopy based on the skin snip biopsies. There are, and then we do some PCRs at the reference lab. RDTs, different types, but they're not the mainstay at the health facilities as well. I mentioned scabies already. For yours, there are two levels. For the field, the DPP test is the main confirmatory test that is done in the field, followed after the initial RDT test, and there's PCR that is done at the reference lab level. This summarizes the tests, the diagnostic tests that currently are ongoing in the country, and in comparison with what WHO is recommending. So Ghana pretty very much is trying to do as many of them as possible. As the only issue now is the scope, okay, the scope of it. Now I'll share with you some key gaps in skin-NTD diagnosis currently, and I will look at these gaps first at the pre-diagnosis phase and at the stage the patient goes to receive formal diagnosis. So at the pre-diagnosis phase, there is a perception based on works that we have done qualitatively that many patients feel that going to the health facility for care can be expensive, and also feel that it takes a long time to receive diagnosis. Many wish that they will just walk in and they'll be told what the issue is. And so when they have to have samples sent to the lab, wait for the results to come back, feels like a long time. The cost is both direct and indirect in terms of the transportation to go to the place, the number of times they have to go to the facility to receive the diagnosis, are all considered expensive when they compare this with other multiple options that they have in terms of the alternative places like going for herbal treatment or traditional care. So the issue of the multiple options are also there that play out. And then also at the health facility, they also have to battle with some barriers when it comes to diagnosis. The first is that many staff across the health levels do not have the confidence as well as the logistics to diagnose. So the confidence has to do with the fact that in a lot of the skin-NTDs, especially take some like Buruli ulcer, lymphatic filariasis, even yours in some places are not very common, are not seen very frequently. So several health workers don't have sufficient exposure with some of these, and therefore that impacts on the level of confidence where the point of care test is available. But in many cases, like I showed earlier, they have to do molecular diagnosis, and this is available only from expert reference labs, and that leads to both logistical and communication bottlenecks. across the chain, okay, across the different cadre of staff and also facilities. So the way forward for skin-NTD diagnosis, as I observed and reflected from what we have done in the country so far, is that one, there needs to be a holistic view on the diagnostic pathway of skin-NTDs, ensuring that we are connecting diagnosis with treatment and support, so that once people are brought closer to diagnosis, they also are brought close to treatment and support to ensure that it's beneficial to the patient. Okay, that's one of the ways that I would advocate that we view the approach to breaking the barriers, to ensure these barriers that are broken links patients to care, which is sometimes a challenge from what we have seen. And in some of our studies, we have to make sure that the studies provide the medications and treatment for the patient, because we see a clear issue where we have improved diagnosis on one hand, and then the care or the treatment support was not available. So that has to be taken care of. So a holistic view of the diagnostic pathway of skin-NTDs will be a way to go. Secondly, we need to also look at integrated strategies for the diagnosis. Several of the skin-NTDs, as we all know, share common characteristics. Okay, they all require detection through clinical examination. In terms of the case management, a lot of them have those in common, and therefore there is the clear argument, which is not in dispute, that going integrated approach would lead to greater cost effectiveness. And this integration across NTDs can also be put together with mainstreaming, which involves ensure the national health systems align to ensure that this integration happens, and also involving the different stakeholders, okay, in the diagnosis pathway. That is something that is being done in Ghana. Of course, there is always room for improvement. I also acknowledge there's a need for more of the point of care testing and multiplex platforms. But with the multiplex platforms, I would say that with a focus, obviously, of bringing the diagnosis closer to the patients. On the screen here, I share with you some target profile, product profiles developed by the Diagnostic Technical Advisory Group of the WHO, looking at some use cases for several of the diseases if new products are to be developed for diagnosis. And you see that there's a lot of focus on diagnosis at the primary healthcare level. There's also a lot of focus on developing point of care tests, and there's also aspects that are experiencing gaps that are being promoted to be addressed. And therefore, I will advocate clearly, without any doubt, that there's a need for more point of care tests. If it is something like Buruli ulcer, we still need point of care testing for them, and several of the ones that are listed up here. In addition to that, also, there's a need for more training for frontline health workers, using both in-person as well as online training approaches. We have done both, and we find it to be useful. Of course, the in-person gives you one-on-one engagement with the frontline workers, but beyond that, you also wanted to have continuous access to the training. And so that is something that should be added to the development of the point of care and multiplex platforms, continuous training of the health workers, as we have found to be very useful. And regarding the online courses, several of them are available, and I show these being promoted by the WHO and available at no cost. And so I encourage people to take advantage of this as much as possible. There's also the skin-NTD manual and app that can be used as a readily available resource for referencing, and also being up to date with the skills that are required to identify various skin conditions of the skin. So in conclusion, I will mention that clearly from the work we've done in the country, Ghana has 7 out of 11 skin-NTDs, and we have majority of them. So you are at the right place. A lot of progress has been made regarding skin-NTD case identification through various research activities, as well as work by the National NTD Control Program and the Ghana Health Service in general. But there's clearly a need for more point-of-care tests and multiplex platforms for diagnosis. Integration, mainstreaming, coordination by different stakeholders are key to meeting the roadmap set by WHO, but I would not stop without saying that all of these will come at a cost, and there's a need to ensure that we put our money where our needs are. In several of the areas we've worked on skin-NTDs, what is very clear is that the skin-NTDs, especially the ulcerated skin-NTDs, tend to be in low numbers compared with other skin conditions that are seen in the area. And therefore, there's a strong advocacy to ensure that we invest in skin care in general, and then in that process are able to target and identify and address the skin-NTDs in the areas. This short presentation, obviously, is not just from ideas from myself alone. It's from work that we've done over the years with several collaborators. I'm grateful to the Skin Health Africa Research Program that has done a lot of work in the country as far as skin-NTDs are concerned. Dr. Kingsley Shiedu of WHO and all other partners who have been supportive over the years. Thank you so much, and I look forward to any feedback you may have.
And today my talk will focus on cutaneous leishmaniasis in the MENA region. And before I start, I would like to emphasize that beyond the fact that cutaneous leishmaniasis has a big disease burden, it also causes strong or very significant psychological impact on affected individuals and communities. So early and accurate diagnosis is crucial for patient management. So that's why I will share today some research from our effort, some results from our research efforts to develop novel molecular diagnostics for the sensitive and specific detection of Leishmania parasites in skin samples. So for those who may not be familiar with Leishmaniasis, they are vector-borne parasitic diseases that are caused by flagellated protozoa of the genus Leishmania and transmitted by the bite of infected female sandfly. So the transmission cycle of Leishmaniasis are very complex and still not really fully understood, but we know that dogs and rodents are the main reservoirs, while the anthropomorphic transmission is also confirmed. So over 20 Leishmania species have been identified as infecting human, and depending on the tropism of the species, the disease may evolve into visceral Leishmaniasis, which is fatal if left untreated, or cutaneous Leishmaniasis that affect more than 1 million people each year over 90 countries or worldwide. And the MENA region is considered as one of the most affected area, accounting like 80% of cases in the Eastern Hemisphere. So like other vector-borne diseases, the incidence and prevalence of CL is strongly associated with risk factors like climate change, poverty, and conflict. And cutaneous leishmaniasis in the Middle Eastern and North African region is caused by 3 main Leishmania species, including Leishmania major, Leishmania infantum, and Leishmania tropica. And today I will talk about 3 case examples from Tunisia, Morocco, and Lebanon. So previously in Tunisia, the epidemiologic situation was very clear. We had like infection due to Leishmania major were predominant in the central part of the country, the Leishmania infantum in the north and Leishmania tropica in the south. However, in the last year, we saw like a very clear shift with Leishmania major moving to the north and south, and Leishmania infantum and Leishmania tropica moving to the center of the country. So as a result, we can see the 3 main Leishmania species co-circulating in the same endemic area. So the same for Morocco, the disease is caused by the three Leishmania parasite species, Leishmania major, infantum, and tropica. And the last year, we saw an increase in the number of CL cases alongside spatial spread. So for example, infections due to Leishmania infantum were used to be sporadic in Morocco, but Leishmania infantum vectors were found with high abundance in all bioclimatic area. So Leishmania infantum infections shifted to the endemic mode. So in the other side, the Middle East, wars and continuous displacement disrupted control programs. So we see in the countries affected by these conflicts a sharp rise in the CL cases. This is due to displacement, healthcare system breakdown, and lack of vector control. So for example, in Lebanon, we noticed a sudden increase of CL cases reported, and this was following the Syrian refugees crisis. So within refugees, we identified infection due to Leishmania major and Leishmania tropica, but evidence suggests that also local transmission has also occurred. So regardless the species, Cutaneous Leishmaniasis has a range of clinical manifestation, and it is called the great imitator, as it can mimic like almost all the dermatoses. And but it is now demonstrated that treatment efficiency depends on the infecting species. So that's why it is very important to identify Leishmania parasites in the frame of the routine diagnosis of cutaneous leishmaniasis. But clinical observation and epidemiological data alone are not sufficient to accurately diagnose the disease. And routinely, okay, I try to, yeah, okay. So routinely, the diagnosis of CL is done by microscopy. That could be also complemented by culture, but these two techniques are laborious, time-consuming. They are lack sensitivity, and they are not able to identify Leishmania parasites as all the species have the same morphology. So the most accurate diagnosis is done by molecular tests, and namely PCR-based techniques. So sorry, yeah, okay. So, but the PCR-based techniques are dependent on equipped laboratory with heavy equipment like sequencers, thermocyclers, electrophoresis systems, etc. But it is known that the global burden of NTDs is like affecting with disproportionately the countries with low or middle resources, income countries. And as a result, we can see that health centers lacking resources are where precisely the diagnosis is most needed. So that's why this stresses the need for a cost-effective point-of-care molecular test to improve the management of patients in the middle or in low-setting areas. So, but what exactly a point-of-care test? So, in 2006, the WHO published a set of criteria that they called assured to identify or to define a PUC test. So, for a test to be a PUC, it has to be affordable, sensitive, specific, user-friendly, rapid, equipment-free, and deliverable. So these kind of tests should be a research priority, especially for NTDs who are referred to as a disease of poverty. So the last decade, we witnessed some technological advancement that supported the development of diagnostic technologies that are suitable for implementation in different settings. So we use these technologies to deliver rapid and simple molecular diagnosis tests for concomitant detection and identification of the main Leishmania parasites encountered in MENA, including Leishmania major, Leishmania tropica, and Leishmania infantum. So to deliver such tests, we adopted this strategy. So for test development, we selected and studied DNA target that were selected depending on the test to be developed and the technology to be used. Then we collected data and samples, or well-documented samples, from different study sites, including in Tunisia, Morocco, and Lebanon. And these samples were used for proof of principle and for test performances evaluation. And then we trained health personnel on these developed tests and built networks in order to facilitate the implementation of these tests. So we start by harmonizing data and sample collection protocols. So we used harmonized form to collect data. We also developed a mobile platform for data management and real-time analysis. We also used a standardized sampling protocol and standard protocol for DNA extraction. And for all the study sites, we used the same gold standard test, including microscopy direct examination and PCR R50ITS1. So the first test we developed was based on recombinase polymerase amplification, coupled to a lateral flow immunochromatography assay for the detection and identification of CL Leishmania species. So we were able, by using this test, to deliver a result within 45 minutes, including 40 minutes for the isothermal amplification and 5 minutes for the lateral flow detection. So the first test we developed was a Leishmania major specific test. And when the assay showed reproducible and stable result when we tested it on reference Leishmania strain, we were able to detect as low as 5 parasites. And when we tested it on CL samples, it showed like a sensitivity of 100% and specificity of 88%. So there is 3 samples that were negative with the standard test, showed positive results with our test. But we think that our test may be more sensitive than the standard one. So for the Leishmania specific assay, it was also stable and showing 100% sensitivity and specificity. So for the one specific to Leishmania tropica, and this is the most complicated test to develop because of the genetic variability of the species. So when we tested the assay on CL samples, it reached a sensitivity of 60% and the specificity of 92% with one cross-reactivity with an infection due to Leishmania major. And overall, so the samples collected from Morocco did not react with our Leishmania tropica specific test. So now we are working on screening for new markers, improved markers to set up the Leishmania tropica specific assay. So the second test I would like to talk about today is based on a handheld ultra-fast PCR duplex coupled to applicant detection using a lateral flow immunochromatography for the concomitant detection and identification of the 3 Leishmania species. So using this test, we are able to deliver a result within less than 30 minutes with 18 minutes for the amplification and 10 minutes for the lateral flow detection. So our PCR were set on a handheld or a portable PCR machine, and the idea was to design 2 types of simplex PCR assays. So the first set of simplex PCR assays is reacting with Leishmania major and Leishmania tropica without reacting with Leishmania infantum. And the second set of simplex PCR assays are reacting with Leishmania infantum and Leishmania tropica without reacting with Leishmania major. And if we put together the two simplex assay and the duplex PCR assay, we'll be able to identify or differentiate between the three Leishmania species. So on the lateral flow immunoassay, we will see a band on the test line 1 for infection with infection due to Leishmania major, band on test line 2 for infection due to Leishmania infantum, and 2 bands on test line 1 and 2 for infections due to Leishmania tropica. So this assay was tested on a set of Leishmania strain, cultured Leishmania strain, and it was it showed a stable and reproducible profile, and we were able to detect between 0 picogram and 40 picogram of the parasite DNA. So this test was not tested on SIA samples, but we tried to mimic biological samples by spiking human blood with a known number of parasites, and we were able to detect as few as 8 parasites. And we did not notice any cross-reactivity with human DNA. So now we are working on the development of a rapid DNA extraction for parasite DNA, and we are using the SwiftX reagent. And these are preliminary data, but they are very promising results. So we were able to detect by PCR using DNA extracted from by SwiftX 100% of positive slides were positive with the PCR using the DNA extracted by the SwiftX rapid DNA extraction kit. And among the negative slides, we were also able to detect 3 slides or 3 samples that were negative with the direct examination, but were positive with PCR that are using the same DNA. So before I finish, I would like to highlight some key messages for the molecular diagnostics development. So target multiplexing has proven essential as it may save cost and time. It also can save samples. And there is one step that we should not overlook, is the step of DNA extraction. So it has to be also rapid and simple in order to facilitate the implementation of the test you are developing. So another key initiative that we already launched is the engagement with stakeholders, especially end users, in order to get their feedback to improve the test in terms of feasibility, usefulness, and performances. So now we organized a workshop to train our collaborators from Tunisia, Morocco, and Lebanon on the RPA lateral flow tests. And we are planning to transfer this technology to the University Hospital of Farhat Hashed Sousse. That is a health center in central Tunisia, and where they receive a great number of SIA cases annually, and test performances in real-life conditions will be soon evaluated. We also organized a hands-on workshop on using mLegionnaire in order to get the feedback of the user and areas of improvement of the application. So finally, we think that dissemination and technology transfer is the key to scale up diagnostic capacity, which is also a key for disease control. So before I finish, I would like to thank my team lab at Institut Pasteur de Tunis, our collaborators at the university hospitals in Tunis and Sousse, Farhat Hashed and La Rabta, our collaborators from Institut Pasteur of Morocco, and the ones from the Faculty of Public Health of the Lebanese University, and finally the Ministry of Higher Education and Research in Tunisia, the National Academy of Sciences in the US, and the USAID. And thank you all for your kind attention.
today that I'm showing you is based on a paper submitted by or presented by Tamaz group of the LMU at the last WHO skin-NTD meeting. And the topic is basically finding new ways for point-of-care diagnostics, molecular diagnostics to diagnose Buruli ulcer, so Mycobacterium ulcerans infections. As a very brief background on the disease, it's a neglected tropical disease, and it was first described end of the 19th century in Uganda. And currently there is around, according to Wikipedia, 3,000 cases reported each year. The disease is caused by a Mycobacterium species called Mycobacterium ulcerans. And those ulcers that you can see here can actually lead to permanent disabilities. And so have the potential to take really a big portion of the quality of life of people who are infected. So, as I said, the presentation is based on a poster presented earlier this year in a WHO global meeting on skin-related NTDs, which is what was prepared by Tamaz, Tsulaia, and his group, and I was one of the co-authors, but obviously you can see I'm the second last, not really important. So nevertheless, since Tamaz wasn't able to make it, I took the chance to present the work that we did here together. And the goal was, as I said, to find, from an extraction point of view, methods or validate methods that are capable to be used also at the point of care. And for that, the methods need to be rather simple, despite the fact that mycobacteria are usually not easy to destroy and the DNA not easy to release. We still tried it with different methods. One of them was SwiftX DNA and other components of our SwiftX toolbox at Xpedite. And for those of you who have never heard of it, I just have this one slide here because the SwiftX way of extraction is quite different from the standard and conventional way of extracting of nucleic acids. We work if we have liquid samples or we have a swab, like from a skin lesion, eluted in a transport medium, then the first step usually is that we utilize magnetic particles and specific buffer conditions to capture those cells without destroying them, capture them, up concentrating them with magnetic beads. So we also don't need a centrifuge for that. And in the next step, after the magnetic concentration and removal of the supernatant, then these cells are lysed, usually by heat. And in the final step, the actual reverse, what we call reverse purification happens, which means that from the lysate that is generated here, these magnetic beads are binding to cell debris and other proteins and other inhibitors in the lysate, in the crude lysate. And by magnetic force, we can separate these out, and what remains is a cleared supernatant that contains the DNA and also sometimes RNA, which can be directly used for a molecular analysis. So for the study that I'm going to present here, we used a couple of those components of this workflow. One of them is the beads that can do capturing of mycobacterial cells and also reverse purification. We tested two different buffer systems which enable the capturing of cells. They are called buffer EN and BPE, you will see it later. And in terms of the cell lysis and extraction, different combinations of buffer DL with and without the help of proteinase K was used, as well as an alkaline lysis approach by this so-called buffer BPL and a neutralization buffer. So the whole setup of the technical validation was like that. Tamaz prepared suspensions of Mycobacterium ulcerans with 4,000 cells plus minus 2,000, so quite accurate. in a PBS suspension, and those were subjected to nucleic acid extraction by different Swiftex variants, by Genolyze as a second point of care suitable method, and also by the reference method that is established at the tropical lab there at the LMU, which is the Gentra PureGene method. And all these extracted nucleic acids have then been detected and analyzed with two different amplification detection methods. One was an IS-2404 specific real-time PCR, and the other one was a more point-of-care suitable isothermal method. So they used LAMP, a LAMP assay, which also detected this IS-2404 element, which is a highly repetitive element in the Mycobacterium genome. So this overwhelming slide here is one graphic of the poster. I tried to break it a little bit down so you better understand what we have done here. So this, what you see here with the red frame, is the PureGene method. This is usually a centrifugation, then lysis for many hours, and then a complicated purification step, as you know it from other standard bind wash elution methods. And then also very long hydration step to end up with the purified DNA. So the blue frame is the Genolyze method. It also utilizes centrifugation for cell enrichment, cell capturing. And then it's a simple two-step process. First an alkaline heat lysis and then a neutralization. From our off-the-shelf products here at Xpedite Diagnostics, Tamaz tested Swiftex DNA, which uses the beads A, which I mentioned, and a certain capture buffer to magnetically capture and enrich mycobacterial cells. And this is this first step here, so no centrifugation in this case. And as a second step, it's a certain lysis buffer that we use, and a heat lysis with and without proteinase K. And at the end, this reverse purification where you just put the lysate into a magnetic rack to clear the lysate off, to remove cell debris and stuff. Then the other off-the-shelf product is Swiftex Parabact. It has a different capturing buffer, which is more sensitive if it comes to capturing of bacterial cells. And it also has a different lysis buffer. As I mentioned, it's an alkaline lysis buffer similar to the one that Genolyze uses. So it's the same approach. Alkaline lysis with heat, a neutralization, and then in our case, a reverse purification at the end. And then Tamaz also explored different combinations of those, like combining the capturing buffer of one kit with the lysis approach of the other, and vice versa. Also tried to use centrifugation instead of our bead capturing, and then coupled this with different lysis methods. And then what he did then was to use the extracted DNA, and as I said, analyze it with qPCR and with LAMP assays. So the results in brief are, he prepared a total of 200 samples, spiked Mycobacterium ulcerans samples. All of them were tested positive, independent of what method had been used for the extraction. That's the case for the qPCR assay, and the same holds true for the LAMP assay. So, and to break this down a little more, this is a graphic of the poster. So here in blue, you see this PureGene method, the very lengthy, old-fashioned way, I would say, of extracting nucleic acids. It has a certain average bacterial count, more or less what he put in. I told you it's like 4,000 copies or cells of the Mycobacterium per sample. So this is what the PureGene also got out, in basically back calculated from the DNA amount that has been detected in the PCR. And then you see here the results in box plots of the different methods. So Genolyze is here as well. In the first glance, you see there is not really a big difference, because if you consider the PCR signal that you get from 2,000 cells, is just 1 CT away from the signal that you get with 4000. So overall, all these methods perform really well. There's not much statistical difference between them, but I want to highlight, of course, the different, or show you how the different SwiftX approaches worked. So SwiftX DNA as the standard kit had a similar performance, and so bacterial DNA yield as the pure gene reference, and also the use of proteinase K for the heat lysis step was not bringing such a huge advantage. It's rather small, but you can see here as well that the cell capturing that we do with the magnetic beads is at least as efficient as a centrifugation. So SwiftX Parabact, similar, slightly less performance, I would say, but not really significant. Then here you see these other different combinations of things. This here is, for example, the centrifugation with a DL buffer, centrifugation with the alkaline lysis. But what actually turned out to be the best is the combination of the capture buffer that is contained in the Parabact kit, the one that is really better for bacterial capture, combined with a DL lysis buffer, which is part of the SwiftX DNA kit, which is one of our standard kits. And that had really a very nice performance, also slightly above the yield in terms of DNA compared to the pure gene method. So to summarize this in one table, for each of these different extraction approaches, Tamaz prepared 20 replicates, so 20 biological replicates were analyzed. And for each of those SwiftX variants, there were 20 of 20 also positively detected in PCR and LAMP. Same holds true for genolyze and also for pure gene. So far, so boring, basically. I mean, more or less expected. The interesting things come below here. If you look at the time, the current reference method is super long. It takes like 12 hours for the whole process because there are very, very long incubation steps in between. And it's much shorter with the genolyze method, which is just under 30 minutes. And even a little bit more, a little bit faster is the SwiftX protocols, which use like 22 minutes for the workflow that he used. Also in terms of robustness, this two processes here, these point of care suitable extractions like SwiftX and genolyze are very, very simple and rapid. The big advantage of the SwiftX is it doesn't need any centrifugation and it doesn't need a neutralization step in most of the protocols. Whereas, yeah, obviously pure gene is very, very lengthy and it has a lot of work steps in between. Is a method that you can do in a central lab, but is nothing that you would do, which you want to do in a field. Also, the technical requirements are very simplified with SwiftX. It's just a heat block and a magnetic rack. You don't need any more equipment. Of course, a pipette, yes, but that's it in terms of instruments. For the genolyze, it's a heat block and a centrifuge. For the pure gene, it's again heat block and centrifuge, but also it needs to work for a couple of the work steps with ice. So you would also need to have access to ice. That means, to summarize, SwiftX method and genolyze showed equivalent analytical sensitivity compared to the reference extraction, which is the pure gene method. Both thus enable a Mycobacterium extraction in less than 30 minutes. And last but not least, the SwiftX shows the highest field friendliness because the instrument needs is the lowest, time is the shortest. Yeah, and so we also have shown with a couple of collaborators that you really can do this in the field in a so-called suitcase laboratory. And that basically is concluding that these SwiftX extraction approaches, as it was also described by Insaf, is really a field-friendly and point-of-care suitable method in itself. which can then be coupled with different analytical methods like LAMP, like RPA, but also everything else. So that is the end of my short process slide deck. I want to thank you and hand over to the moderator, and I'm happy to answer your questions.